Alrighty, we left off here. I'm a little farther away from the screen because if I move closer, you can see the sun setting and it lights me up plenty. We left off at metaphase, and you learned at metaphase that all of the chromosomes that are sister chromatids at this point, they are arranged as X's right there in the middle. And the reason they are is because spindle fibers from the spindle apparatus, which in animal cells includes the centrioles, which are also called, which in pairs are called centrosomes, too many centros, are tugging them so hard. As a matter of fact, they tug them so hard that poof, they pop apart. And as soon as they pop apart, you can see that they go to opposite sides of the cell. Let me just make me out of the way. Okay. So the X of a replicated chromosomes, sister chromatids, and they get popped apart, and now they're traveling to opposite ends of the cell. I will point them out right here. So there and there. And you can see somewhere in there, this is an animal cell, somewhere in there, there are two of these centrioles. Together they make a centrosome, which in animal cells is parts of the spindle apparatus. The spindle fibers are another part of the spindle apparatus. And in animal cells, they're tugging the individual chromosomes to opposite ends of the cell. Then we've got telophase. And in telophase, the nuclear envelope starts to reorganize, and you can still see condensed chromosomes, but they're not usually organized as neatly. Let me give you a hint. When we're showing you pictures that we want you to identify in Bio 120, usually telophase is going to be in animal cells, and you will see the development of what is known as a cleavage furrow. Cleavage furrow. Let me write that for you. A cleavage furrow. Cleavage furrow is that area right here, bing, right there, bing, where you can tell that the cell is being pinched in half, almost like a string is being drawn around it to pinch it in half. If you see that stage in animal cells, then you will know that we are now in telophase. Right? Does telophase have to happen with cytokinesis? Technically, no. Telophase is part of mitosis. Cytokinesis is actually the word for cell division. Now, a little bit about cytokinesis or cell division in plant cells. Plant cells have got a cell wall, right? In animal cells, you will get this cleavage furrow. You'll get a cleavage furrow in animal cells. That's the cleavage furrow, right? But a plant cell has got this shoebox arrangement around it. So a plant cell cannot do a cleavage furrow. That makes plant cell division more difficult. And that is a question on next week's quiz. Why is plant cell mitosis, at least cytokinesis, more difficult? And the reason is that plant cells can't do that pinching in half. They actually have to build a new cell wall between the daughter cells. Okay? All right, let's practice a little bit. I keep having to move me out of the way. All right, let's practice. So look at all of these cells here as if you're looking through a microscope and tell me, which one do you think is in metaphase? I like to pick metaphase first because it's kind of an easy one, right? This one's in metaphase. They're all lined up there at the middle of the cell. Great, okay. Which one of these cells would you imagine is in anaphase? So remember, it's PMAT, metaphase, everything's lined up at the middle. Okay, so after they're lined up at the middle, they're doing this. Which one of them's doing that? This one. This one is anaphase, right? right? So A for anaphase. Which one is in telophase? I don't see a telophase in this one. No. Which one of these is in prophase? In prophase, the nuclear envelope goes away and the chromatin goes from just looking like a bowl of spaghetti to condensing. This one is in prophase. Alrighty, that's it. Watch the Amoeba Sisters videos, do all of your lab activities because those are important for points. And also, if you don't complete them, it means you haven't attended this week. And that's reason for you to be dropped from the class. And I'll see you next week.